Hello students. So we shall continue with our concept. So the next one is the convention for naming restriction enzymes. So already we have discussed the restriction enzymes. So these are the enzymes that restrict the growth of virus. Because of this, they are called restriction enzymes. The types of restriction enzymes we shall consider after some time. So first we shall consider the convention, right? Convention that is used for naming restriction enzymes. Now what is this convention? So convention is the, you can say, system, right? Convention is a system that is used for naming restriction enzymes. So what is the system that is followed for naming these enzymes? So here first we shall consider one example of restriction enzyme. So we can consider any example. So here I will consider we shall consider the example here as ECOR1. So this is an restriction enzyme. As you know, these enzymes, they are discovered from bacteria. So restriction enzymes are discovered or they are isolated from bacteria, right? So this is one example of restriction enzyme. So what is the convention, right? What is the convention that is used for naming this particular enzyme? So here the first letter that is capital E. Now this capital E is the genus. Capital E is the genus of this bacterium or generic name of this bacterium. So here the generic name of this bacterium is S. escherichia. So S. escherichia is the name of the genus or generic name, which can be abbreviated to E. So escherichia can be abbreviated to E. So that is the first letter, right? Then CO. What is this? CO. CO is the specific name or species. CO is the specific name or species of this bacterium. So here CO, it stands for coli, right? CO stands for coli. So this enzyme ECOR1 is obtained from a bacterium called, now which bacterium? So it is obtained from a bacterium called E. coli, right? So from E. coli bacterium this restriction enzyme is isolated. Then the fourth letter, capital R. Fourth letter, capital R. It is the strain of this bacteria, right? Capital letter R is the strain of this bacterium. Now what do you mean by strain? Strain, it can be called as the type, right? So capital R indicates a particular type of this bacterium or strain of this bacterium. So that strain is RY13. So RY13 is the strain of this bacterium. After that, the last one is the Roman number. So it is the Roman 1, right? So Roman 1 is the, we can say, sequence in which this enzyme was isolated from this bacterium, right? So 1 or Roman 1 is the sequence. It is the sequence in which this enzyme was isolated from this bacterium. So we can say this was the first enzyme isolated from this strain of bacteria. That's why this enzyme is called ECOR1. So understood this? So this is the convention that is followed while naming restriction enzymes. So E is the generic name, that is Escherichia. CO, it is the specific name or species, which is coli. R is the strain of this bacterium, that is RY13. Roman 1 is the, you can say, Roman 1 indicates the sequence in which this enzyme was isolated from this bacterium. So here it is the first enzyme that was isolated from this bacterium. So that is the convention followed. So you may expect this as a 2 or 3 mark question. Mention the convention or write the convention for naming restriction enzymes. Fine. So after this we shall consider the next part, that is so the next part is the classification of restriction enzymes. So restriction enzymes, they belong to a larger group of enzymes which are called nucleases. So restriction enzymes, they belong to a larger group of enzymes which are called nucleases. Now these nucleases, they are classified into 2 categories. So they are classified into 2 categories as exonuclease and endonuclease. So nucleases are classified into 2 groups as exonuclease and endonuclease. Plural, exonucleases and endonucleases. The example for exonuclease, the example here is Exo1 is the example of exonuclease. The examples for endonuclease, so this is the example of endonuclease. Even Hind2, it is also an example of endonuclease. Now, what are these exo and endonucleases? See, nucleases are a larger group of enzymes. Exo and endo, these are the opposite terms. Exo and endo are opposite terms, right? Just like out and in, right? So here exo indicates out, that is external. Endo indicates in, that is internal. So exo means out or exterior. Endo means in or interior, right? So these enzymes, that is nucleases, they can cut the DNA or they can remove nucleotides. They can cut the DNA or they can remove nucleotides from DNA. Already I have studied nucleotides. So nucleotides, these are the products formed by linking nitrogen base, sugar and phosphate. When nitrogen base, sugar and phosphate, when they are linked together, you get a product that is called nucleotide. And DNA, it is made up of a chain of nucleotides linked by phosphodiester bonds. Right? Already I have studied this in the previous chapters. So exonuclease, it is an enzyme that removes the nucleotides of DNA from the sites. So exonuclease removes the nucleotides from the sites. That's why it is called exonuclease. So in simple words, it cuts the DNA from the sites and the nucleotides are removed. Exactly opposite is the endonuclease. So what is this endonuclease now? So endonuclease cuts the DNA at a particular region, right, within the DNA. So here, this enzyme, it cuts at a particular region within the DNA, means it cuts the DNA from the somewhere from the center. Within the DNA means somewhere from the center. So it cuts the DNA at a specific point. What is that specific point? Right? Endonuclease cuts the DNA at a specific point. That specific point, it is called, we have discussed that in the previous video. It is called recognition site. It is called recognition site. What are the other names for recognition site? It is also called restriction site or recognition sequence or palindrome sequence, right? So the nucleases which act at a specific site within the DNA, right? They cut at a specific point within the DNA. It is called endonuclease. And the examples of endonucleases. So one is here, ECOR1, then HIN behind 2. Right? Those are some examples. Other few examples we shall consider in the coming videos. So that is about the classification of restriction enzymes. Then, so here you may expect some questions. Classify the restriction enzymes, right? Or what are nucleases? Classify the nucleases. So you may expect this as a 1, 2 or 3 mark question. So we shall continue with the next concept, that is application of REN, that is application of restriction endonuclease. So what is the application of endonuclease? So here the meaning of application. What is the meaning of application? Use. So what is the use of endonuclease or restriction endonuclease? It is used in genetic engineering, right? Restriction endonuclease is used in genetic engineering. That is the main application of REN. It is used in genetic engineering for what purpose? For constructing recombinant DNA. So to construct recombinant DNA, REN is used. So how REN is involved in the construction of recombinant DNA? See here, this REN, it is a molecular scissor, or we can call it as molecular scissors. So this REN cuts the DNA, right? That is, it cuts the plasmid. It cuts the plasmid. Even it is used to cut desired gene. So plasmid and desired gene, they are cut by using REN. And after this, that desired gene is inserted in the plasmid by using another enzyme, that is DNA ligase, and ultimately you get a molecule that is called recombinant DNA molecule. So this is the application of restriction endonuclease. So it is useful in genetic engineering for the construction of recombinant DNA. Where REN cuts, it is used to cut the plasmid and desired gene, where both the plasmid and desired gene, they are linked by ligase to produce recombinant DNA. The next we will consider so in the construction of recombinant DNA, the REN, for the construction of recombinant DNA, the REN which is used, it should be of same type. Same type REN has to be used to construct recombinant DNA. It means, see here, for constructing REN, a plasmid and desired gene are used. A closed plasmid has to be made open, that is with the help of REN. Desired gene is isolated from DNA, that is with the help of REN. So same REN has to be used, or same restriction endonuclease has to be used for cutting plasmid and for isolating desired gene. Right? Why same REN should be used to cut the plasmid and to isolate desired gene? The reason is, when same REN is used, similar sticky ends are produced. Right? When same REN is used, what happens? When same REN is used, similar sticky ends are produced. When the sticky ends are similar, what are the sticky ends? We discussed already. So sticky ends are single strands of DNA which are complementary, right, to the desired gene. So sticky ends, they are single strands which favor formation of hydrogen bonds with complementary strands, and even they facilitate the action of enzyme DNA ligase, right? So because of this, you can easily construct the recombinant DNA if same REN is used. Imagine if different restriction endonucleases are used. For example, if you cut the plasmid with hind 2, and if you cut or if you isolate desired gene with ECOR1, so here two different REMs are used, one for cutting the plasmid, another one for isolating desired gene. So what will happen if different REMs are used in the construction of recombinant DNA? What will happen? Dissimilar sticky ends are formed. So the sticky ends which are produced on the plasmid and on the desired gene, they are not same. So dissimilar sticky ends are produced, and these dissimilar sticky ends, what is the disadvantage? They will not facilitate the action of ligase, right? It will not favor the formation of hydrogen bonds, and it will not facilitate the action of DNA ligase. So because of this, you cannot construct recombinant DNA, right? So here we may expect some questions. Why same REM is used to cut plasmid and to isolate desired gene? Fine. So like this, you can expect certain questions on this particular concept. So with this, we shall conclude and we shall continue in the next video. Thank you.